Hello, my name is Melissa Cross. This is going to be a presentation on the structure and function of a single synapse. So let's start at the beginning with neuron cells. So neuron cells are actually nerve cells that are signaling pathways for the nervous system. So the synapses are located at the end of the axon terminal, right here. And as you can see, there are many of them coming off the end of the neuron cell. These axon terminals, or the synapses, butt up against target cells such as blood vessels, cardiac cells, and muscle cells. So they're basically taking a package signal and sending it out to other cells in the body. The main functions of these cells is to send signals throughout the body by means of chemical, electrical, and hormonal signaling. As you can see, the direction of the impulse follows this arrow. So nerve cells are different than any other types of cells in the body because there are many different parts. First, we have this dendrite, which is going to be the receiving end of signals for this cell. So there can be other axon terminals butted up against this dendrite, and it's going to receive a cell, and it's going to send it out this way. These cells do have nuclei. Obviously, they're cells like any other cell. They just look a little different. And we have the soma, which is the cell body. The signal gets sent through this long chain called the axon, and it is protected by the myelin sheath, which is a lipid layer that protects the signal going through the cell. So there are two different types of synapses. There are chemical synapses and there are electrical synapses. Chemical synapses are more common and widely known. There are many differences between both, as shown in the chart, and we'll dive deeper into those differences on the next slides. But some important things to note about each are if the synapses are physically connected, how the signal is transmitted between synapses, such as how fast, what types of particles go through, as well as how fast these signals travel. So we're going to first start with electrical synapses. So first let's go over some of the structures located in this picture. We have the presynaptic cell, which is the cell at the top of the picture, and this cell is going to be sending the signal. At the bottom of our picture, we have the postsynaptic cell. This cell is going to be receiving the signal. We also have something called the gap junction. So this gap junction allows both cells, the presynaptic and postsynaptic synapses, to be physically connected. This allows small charged particles, such as ions, through without the particle ever having to touch the extracellular fluid. So this allows for an almost instantaneous sending of the signal, sending and receiving. So, like I said, electrical synapses are physically connected via the gap junction, which is made up of ion channels that allow the cells to directly communicate. Electrical synapses are less common than chemical synapses and work best where a synchronization of activity is needed. For example, in muscle contractions and cardiac contractions, where all the cells in that organ need to contract at the same time. Because of this synchronization, these synapses are typically either firing or not firing all at once and are typically unregulated. So for example, there is a cluster of neurons in the brain that all secrete the same neurohormone involved in the reproductive system. Because of the electrical synapses, these neurons all fire in synchrony, so at the same time, resulting in coordinated bursts of secretion once every 2 to 3 hours, with no secretions occurring between these bursts. And this is actually noted in our textbook. So the ions will flow from the presynaptic cell through the gap junctions, transmitting those signals, and they're actually allowed to move back and forth between the gap junction. This is a bidirectional movement from pre to post. There is no conversion of signaling from chemical to electrical, such as in the chemical synapse, which we'll get to in the next slide, which allows, again, these signals to be transmitted virtually instantaneously. Now we're going to talk about the function and structure of chemical synapses. So we're going to quickly go over some of the structures involved. This one is a little bit more involved than the chemical synapse. So again, we have our presynaptic cell, which is sending our signal. We have our postsynaptic cell, which is receiving our signal. And this time on the presynaptic cell, we have something called a voltage-gated calcium channel. So this takes in calcium ions and creates a more positive environment inside the presynaptic cell. Inside the presynaptic cell, we also have the synaptic vesicles. So these synaptic vesicles are just little transport bubbles for the neurotransmitter to make it easier for them to go through to the other cell. Neurotransmitters are actually chemical messengers packaged in the presynaptic cell. They're going to transmit the information from the neuron to the target cell or to another neuron. On the postsynaptic cell, we have the receptor for our neurotransmitters, also called the ligand-gated ion channel. In this case, the ligand is the neurotransmitter. And then something very important that is a difference between electrical and chemical synapses. In the chemical synapse, there is a synaptic cleft. So this is the two cells, the presynaptic and the postsynaptic cell, are not touching at all. There is a juncture between the two where the neurotransmitter is going to flow between. So, like I said, the synapses are separated by the synaptic cleft. Once the calcium creates a positive influx by going through the voltage-gated calcium channel, the neuron allows the vesicles to move toward the synaptic cleft and release the neurotransmitters between the synapses. The ligand-gated ion channels can only accept the specific neurotransmitter being transported to the second synapse. The neurotransmitters binding the receptors in turn create an action potential moving the signal to the other cell. The overall function of these synapses is to move the neurotransmitters, in turn creating an electrical signal in the postsynaptic cell. The conversion from chemical to electrical actually causes the signal to take a little bit longer than the electrical signaling. So instead of being instantaneous, we're going to have about a 0.5 to 1 millisecond before that signal can transfer. And the conversion also causes the signal to be unidirectional, so only moving in one direction. So we have these neurotransmitters only moving in this one direction. They cannot move back this way. Once received by the ligand-gated channels, the neurotransmitters are going to bind, and then they're going to create an electrical signal that is passed through the postsynaptic cell. These are my references. I used Khan Academy for my pictures, as well as watching a video on some of the functions of synapses. And I used our textbook. And yeah, thank you so much. Hope you learned a lot.